Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on types of DNA. So, variations present in the conformations of the nucleotides of the DNA that produce different types of DNA. So, variations in the conformations of the nucleotides. So, DNA is a polymer of nucleotide, you know that. If the variations present in the conformation of nucleotide DNA sequences, so that produce a conformational change that yields different types of DNA. Okay, so there are four types of DNA. Let's see one by one. First one is bDNA. Second one is aDNA. Third one is zDNA. And last one is cDNA. These are the four types of DNA molecules they produced due to the conformational change in their nucleotide sequences. Okay, so let's see one by one. Okay, so let's see the first DNA type, that is B type DNA. So bDNA is nothing but it is commonly found in all cells of the organisms. Okay, it is commonly present in all the cells of the organisms. So it is showing a right-handed helix. So it is right-handed helix or clockwise manner. It is showing the helical structure in a clockwise manner. Okay, so and also 10 base pairs are present per helix. So 10 base pairs are arranged per helix. So it is a right-handed helix. It is arranged in a right-handed helix manner or clockwise. Okay. So 10 base pairs are arranged per helix. And the pitch of the helix is 34 angstroms. Pitch is nothing but the distance or the length of the helix is nothing but pitch. Okay, the length of the helix is nothing but pitch, that is 34 angstroms. The distance between two base pairs is 3.4 angstroms. The distance between two base pairs is 3.4 angstroms. So, let's see the second type of DNA, that is aDNA. aDNA is nothing but dehydrated DNA. aDNA is a dehydrated DNA. That means dry DNA. Actually, we have studied in the previous video, Wilkins and Rosalind, they got pure form of DNA in the form of photographs. Okay, so they get two types of DNA. One is wet type DNA, second one is dry type DNA. So wet type DNA is nothing but bDNA. We have studied the complete structure of DNA in the previous video. So second one, they got Rosalind and Franklin, they got second type of DNA, that is nothing but aDNA, that is dehydrated form. That is in dehydrated form, nothing but dry DNA. Okay, so it has 11 base pairs per helix. It has 11 base pairs per helix. Okay, and it has a pitch of 28 angstroms. The length of the helix is nothing but 28 angstroms. So it is also a right-handed helix. It is also showing right-handed helix. Okay. Let's see the next type, that is nothing but zDNA. Z is nothing but zigzag DNA. Okay. So zDNA is a left-handed helix. It is showing anti-clockwise helical structure. First time I am telling anti-clockwise or left-handed helix. Okay, remember. So it has 12 base pairs. It is a left-handed helix. It is showing left-handed helix. So it has 12 base pairs per helix. 12 base pairs per helix. It has the pitch of 45 angstroms. That means the length of the helix is 45 angstroms. So in zDNA, the sugar phosphate backbone of DNA that follows a zigzag manner. Okay, sugar phosphate backbone that follows in a zigzag manner. So that's why it is called as zDNA. That's why the name has given as zDNA. Okay, and also it have a single groove with a greater density of negative charges when compared to the bDNA. Okay, it has a greater density of negative charges than that of bDNA. Okay, so actually zDNA is reported only in vitro conditions. It was not reported in vivo conditions. Okay, it is only reported in vitro conditions. Okay, so let's see the next type, that is cDNA. So cDNA is also a right-handed helix. It is also showing right-handed helix or clockwise manner. So it has 9.33 base pairs per helix. It has the diameter of 19 angstroms. It has the pitch, nothing but helical length, that is 31 angstroms. But it is more tightly packed than the bDNA. Okay, so let's see the comparison between different forms of DNA. So there are 4 types of DNA. So let's see the comparison. First one is helical sense. bDNA is a right-handed helix. It shows the right-handed helix. ADNA too showing, ADNA also showing right-handed helix. Only zDNA which is showing left-handed helix. cDNA also showing right-handed helix. Let's see the pitch. Pitch is nothing but the length of the DNA helix. So bDNA consists 34 angstroms pitch. So ADNA consists 28 angstroms pitch. So the wider pitch present in zDNA, that is 45 angstroms. cDNA consists 31 angstroms pitch. Okay. So let's see the number of base pairs per helix. So number of base pairs per helix. Here 10 base pairs are present per helix. Here 11 base pairs are present per helix. In zDNA there are 12 base pairs present. In the case of cDNA, 9.33 base pairs present per helix. Okay. So let's see the diameter of the DNA helix. Okay, diameter. So bDNA having a diameter of 20 angstroms, ADNA having a diameter of 23 angstroms, whereas zDNA having a diameter of 18 angstroms, cDNA having a diameter of 19 angstroms. Okay, so let's see the distance between the base pairs. Distance between the base pairs. 3.4 angstroms. Distance present between the base pairs. 2.56 angstroms. In case of zDNA, 5.71 angstroms. Whereas in the case of cDNA, 3.32 angstroms. Okay. So let's see the arrangement of sugar phosphate backbone. So they are regularly arranged in bDNA. Here also regular arrangement observed in ADNA. But in the case of zDNA, the arrangement is zigzag, irregular arrangement. Simply you can say zigzag arrangement. Okay. So in cDNA also the arrangement of sugar phosphate backbone is regular type. Okay, students. So these are the differences between different forms of DNA. So let's see the functions of DNA. Okay. So let's see the functions of DNA. So DNA stores all the genetic information. So it stores genetic information. So the DNA stores all the genetic information or instructions that are needed for growth, development, functioning and also reproduction of all living organisms. Okay. So it stores the genetic information which is needed for the growth, development, functioning and reproduction. So second one is transmission of heredity information. So the DNA, it carries all heredity information. The information present in an organism that should be shared by the organism to their offspring. The heredity characters, they are transferred from the parent generation to the offspring. Okay, so actually gametes, they receive genetic information. When they combine to each other, it will result in the formation of an individual. The individual getting all the genetic information from their parents only. So the DNA passes genetic information from one generation to the next generation. Okay, next one is protein synthesis control. DNA provides all the information which is necessary for protein synthesis. Actually, it involves two steps: transcription and translation. During the transcription, the information present on DNA, which is transcribed to mRNA. In the translation, the information present on mRNA in the form of codons, that is translated into a protein. So proteins are necessary biomolecules for the structure and functioning of particular organism. So let's see the next function, that is replication. The DNA can make an exact copy of itself. That means during the cell division, the parental cell has to provide the genetic information, nothing but DNA, has to share this DNA to two daughter cells. Okay, so that's why it should undergo a self reproduction during the S phase, nothing but synthesis phase. So there the one DNA molecule can reproduce into two molecules of DNA. So the parental cell will share these two strands of DNA to two daughter cells equally. So this process is called as autocatalysis. Autocatalysis or autocatalysis. Okay. Let's see the next function. Regulation of cellular activities. All cellular activities, they require enzymes and proteins. So they were just prepared by using the information present on DNA. Let's see the next function, that is mutation and evolution. So, if any sudden changes occurs in the DNA sequences that leads to the variations in the traits. So, these mutations or variations, they are only responsible for the evolution and natural selection. Okay, so the sudden changes occurs in sequences of DNA that leads to the mutation. Those mutations, they are responsible for the variations. They involved in the evolution and also natural selection. Okay, so this is about the types of DNA and the functions of DNA, students. If you like the video, please subscribe my channel. Thank you.